Genome Center, also using it for population biology, and also in the microbiology and immunology and still in medicine. He was very instrumental in inspiring us to take on this project.
I lied to them a lot.
You did, actually.
He was very interested in microbes and got us excited about it, and today he's going to speak to us about DNA and the world of microbes.
And I have a cold, so apologies here if my voice comes in and out, and I'm sipping lots of tea while I do this. So what I thought I should do before the real speakers come out here is give a little bit of background, because I figured that the audience was not going to be completely microbophilic in some way, or familiar with the sort of background behind what the research that a lot of the people here are doing, and to save some of the speakers the necessary steps from giving some of that background. What I'm going to do is just basically give a brief tour of the history and the reasons behind why a lot of people are using DNA-based studies to study microorganisms. And please stop me anywhere along the way. I actually like to do chalk talks more now than slide talks, although I kind of had a panic with my voice that I needed slides to just give props while I couldn't speak. What I'm about to tell you first is what drives me, and I won't spend a lot of time talking about a lot of my research, but I wanted to give a little bit of background to what I'm interested in terms of microbial diversity. So I'm a recovering birder. My first field biology work was on hummingbirds out in Colorado, and I grew up getting interested in natural history, studying birds. And I love this story. There was an American robin that showed up in London, of all places. And instantly, the Twitterers, as they're called there before that computer program came out, that follow all the birds, some people noticed this and posted announcements to various birder message groups that there was this American robin, and immediately everybody sort of who was fascinated by birds in England, which, you know, there's not much other wildlife there, so they kind of glommed on to go look at this American robin and proceeded to watch a falcon eat it in the next day or two. But the reason I want to tell you about this is how this could even happen. And the reason this could really happen is that birders have compiled information over many, many, many years, centuries even, about the different kinds of birds, where those birds are found, how to identify those birds, the properties of those birds, sort of the historical biogeography, the temporal variation in the patterns of those birds. And I have like 4 copies of this bird book. One's in my car, one's in my garage, one's in my Genome Center office, and I think there's one in my Evolutionary Ecology office. And it's great because this is like the sum knowledge of all the information that you would have on sort of where you would expect to find and how to identify birds. This is what I want. I want the same thing for microbes. I want to be able to know when we go to a particular place, a niche or an environment or whatever, what microbes we expect to find there. And if we go to some place, say the air in the Senate building in Washington, D.C., which, you know, we had the anniversary of September 11th yesterday. There's going to be an anniversary of, I think it was September 18th when the anthrax letters came out. How would you know if there's a microbe that's not supposed to be there in a particular place? We need a field guide. So there are lots of reasons why a field guide would be useful. If someone has a disease, you'd like to know what the healthy state is versus the disease state. You'd like to know all of this information. And a field guide is sort of a useful concept for representing this. It's information about the phylogenetic diversity of organisms, their biological properties, their biogeography, and their sort of how to identify them, all wrapped into one sort of guide. And that's what my dream is, to build one of these, seriously build one of these for microbes. So I'm going to give a little bit of background on microbes. I assume most people here are not quite as antimicrobial as many of the people we interact with. But you know, the big deal about microbes, of course, is that they're small. And in general, the definition that many people use is organisms that you can't see without some aid, like a microscope or a humongous magnifying glass or something to that effect. And they weren't really, most of them weren't really understood, at least directly, until people started looking in microscopes at samples to characterize the microbes that were present in them. And of course, despite being small, there are a lot of them. So once you get a microscope and you start to count the microbes present in a particular place, you can count a lot. So the estimate is that there are more microbes on the planet than there are stars in the universe. There are hundreds of millions of cells in a small sample of soil or water. If you count viruses in with microbes, which I'm not going to get into the argument about whether or not we should, but if you count viruses, in a small amount of water sample, you can find hundreds of millions of viruses. Whether or not they're functional viruses is not always clear, but there's lots of microbes out there. And the big thing for people studying the human ecosystem is there are, in some estimates, at least 10 times as many microbial cells in and on people as there are human cells. So there are lots of microbes out there. Not surprising, they're small, right? But if you add up their total sort of volume or total weight, they actually contribute a lot to the biomass on the planet. So the total amount of biomass is estimated to be more than that for plants and animals present in microbes. And we microbe fans like to point out that not all microbes are boring when you look at them in the microscope. There's incredible diversity of form of different microorganisms. A lot of that diversity is seen in the microbial eukaryotes, things like diatoms and foraminiferans and algae of various kinds, and incredible diversity of cell shapes and outer cell sort of structures that organisms create. There's also a lot of diversity even in the bacterial and archaeal world, the organisms that don't have nuclei. So this down here is a fruiting body for bacteria. And there are a variety of bacterial species that form multicellular or multicellular-like structures and complicated filament structures, biofilms, all sorts of other things. There is a lot of diversity of form out there in the microbial world. And we get excited about this diversity of form, but funding agencies generally don't care. They're much more interested in the diversity of function that's present in microbes. And of course, a lot of the microbes that people study are the nasty ones. This has historically been, first of all, the easiest to study in some cases because you can study a disease as opposed to the microbe itself. But there's, of course, lots of work on microbes that cause various diseases of plants and animals and other organisms. There are the good microbes. These are ones I'm more interested in. Mutualistic symbionts that work with some other organism where in some sort of tight symbiotic association where both of those organisms benefit. And there are a plethora of examples of these, from nitrogen fixing in the roots of legumes to the chloroplasts that are inside all plants. Those are ancient relic cyanobacteria. Mitochondria were once free-living bacteria a long time ago. And then inside pretty much every plant and animal that people look at, there's some suggestion of some type of mutualistic symbiosis present with microbes. And there are all sorts of bizarre ones, like ones in the bottom of the ocean where there are these giant tube worms. This is how I got interested in microbiology, actually. I was working as an undergraduate with a professor who was studying these giant tube worms in the bottom of the ocean. They have no mouth. They have no digestive system. They basically function like a plant, except instead of chloroplasts, they have chemosynthetic bacteria that are intracellular inside the cells of this worm. And the worm brings in hydrogen sulfide, carbon dioxide, and oxygen for its sort of internal structures. It uses the oxygen, it feeds the bacteria carbon dioxide and hydrogen sulfide, and then the bacteria make sugars as well as all the other things the worms need. And there are lots of these chemosynthetic symbioses found throughout the planet. So there's lots of these sort of mutualistic associations. Some that you will hear about today are what are generally called commensal associations. Formally, commensalism means that there's a symbiosis where two organisms live together and interact, and one of them benefits, and there is no effect on the other. When many people use the term commensalism, what we really mean is we don't know what those organisms are doing in this system. It's not that we've shown that they're not a mutualism. It just basically means that there's some type of interaction that's not obviously parasitic or mutualistic. And there are lots of organisms like this living in a cloud, basically in and around plants and animals, generally now called the microbiome, the microbial ecosystem around plants and animals. Some of the ones I'm more interested in are free-living organisms, well, organisms not obviously associated with a large plant or animal host. They may or may not be free-living, that thrive in extreme conditions, and in very extreme environments, they tend to be dominated by microorganisms that are able to survive and thrive in those environments. Obviously, there's lots of people here who are interested in this type of thing. Davis is a good place to study them. There are microbes involved in a lot of agriculture and food production and make lots of the things we think are quite yummy in many ways. Cheeses, bread, beer, wine, fermentation of various products like soy sauce and other things. And, you know, again, the microbial people like to point out that sort of microbes basically run many of the global nutrient cycles on the planet. So they play important roles in all these associated functions, and there's incredible diversity, again, of form and function among the microbes. And so in terms of sort of microbial ecology, this is a condensation of, you know, 200 years of research in microbial ecology to two questions that people ask a lot about microbial ecosystems. Who is out there? That is, what are the organisms out there in some particular environment, like the human gut? And what are they doing? What are the functions of those organisms? And what I'm going to tell you about for the rest of the talk is how sequencing is used as a component of these studies. I don't mean to imply that there aren't other ways to study who is out there and what are they doing, but DNA sequencing has been used very efficiently, and it's getting cheaper and cheaper to do, so there are more and more people using DNA sequencing methods as a surrogate for studying microbes and microbial diversity in general. And what I'm going to do is go through what I call eras of the use of sequencing in studying microbial diversity. And so again, please stop me anywhere along the way if any of this doesn't make sense or you have questions. So what I would consider the first era related to using sequencing to study microbial diversity is what we can call the ribosomal RNA tree of life. So in the 1970s, Carl Woese in particular, as well as a few others, started using very painstaking chemical methods to read small fragments of the sequence of ribosomal RNAs from different organisms. And there were a variety of reasons that they were doing this, but they started to take organisms that they could get a big sample from. For microbes, that was ones that they could grow in the laboratory. Extract the ribosomal RNA from those organisms and run this chemical method to read little tiny fragments, oligonucleotide fragments of the sequences from the ribosomal RNAs from those organisms. And then Woese did a thing which other people weren't quite doing at the time. He started to compare the patterns of those oligonucleotide fragments between different organisms to see what the similarity was between different taxa. And he started to discover things like, for example, that people had classified something as, say, Pseudomonas, when actually many things called Pseudomonas were not particularly closely related to each other. But then he made a much bigger discovery, which was he started to compare the patterns in a lot of different microorganisms in particular, and discovered that instead of one major group of organisms that didn't have a nucleus, the so-called prokaryotes, there actually were two groups of organisms that didn't have a nucleus. And there was this new group that he originally called the archaebacteria, subsequently called the archaea now, that represented a third branch in the tree of life. And that this branch was previously not seen because of the way that people had classified organisms in building a tree, had sort of left out microbes in general and left out organisms without nuclei in particular. And when he used sequence data, even these tiny little fragments of sequence data, he was able to build sort of the first tree of life that accurately or remotely accurately included all organisms on the planet. And so he came out with this three-domain tree that is used generally now in lots of different areas, where there were these three groups: the eukaryotes, the bacteria, and the archaea. Now, lots of people still use ribosomal RNA sequencing for various purposes, and I'll show you some more examples throughout the rest of this. But the big sort of discovery in ribosomal RNA analysis was this discovery of the third branch in the tree. Now, the ribosomal RNA tree is not perfect. There are many reasons why the ribosomal RNA tree of life has some idiosyncrasies that you might not want to use it as your perfect representation of the phylogeny of everything. There are certain ribosomal RNAs between thermophiles that are not closely related, for example, converge on GC content with each other and can create misleading phylogenetic patterns. Many people now, when they try and build big trees of life, try and include protein coding sequence when they do this. So this is a tree I took from Sandy Baldoff, who built a phylogenetic tree of all organisms using protein coding sequences instead of ribosomal RNA. It gives the same general picture, but some of the subtle details, like where particular individual groups go within the bacteria and archaea, is different. But the point is, this first era was phylogenetics, evolutionary classification of microbes based upon sequence data. What you could call the second era of using sequences to study microbial diversity started mostly with someone named Norm Pace, as well as a few other people who took what Woese was doing, and what Woese had focused on mostly was organisms, microbes that you could grow in the lab so you could get enough material to do this ribosomal RNA sequencing. And that turns out to not be give you a complete picture of microbial diversity, and there's something called the Great Plate Count Anomaly, which is sort of commonly cited as an example of this. If you go to an environment, take a sample and split it in two, and you grow the things from half the sample and see how many colonies you can get out of that half, and you look at the other half in a microscope, you see that there are many, many more cells in the microscope than colonies you're able to grow. And this has generally been called the Great Plate Count Anomaly. And there's been a big debate over many years as to what exactly causes this in every environment. But one of the explanations is that some of the organisms in the environment, if not many, cannot be grown currently in culture in the lab. So we miss those when we study only the organisms that you can culture in the lab. And what Pace realized was that you don't have to culture them, you can characterize them through studies of their DNA, of their nucleic acids. And what he actually originally did was go to the environment and extract ribosomal RNA from the environment and read the sequences of ribosomal RNA from the environment. But very shortly after that, I'm just going to skip over. I was going to argue that field studies in plants and animals are kind of fun. You can do field studies in microbes, and many people do them, but they are challenging, and this is why people have turned to DNA to supplement some of those field studies. So what you do is you go to an environment, you collect DNA from the environment or RNA, and you can read the sequence from those samples just like you could from a cultured organism. What revolutionized this area, though, was the invention of PCR, the polymerase chain reaction. And this allowed people to go to environmental samples and specifically amplify out of those samples ribosomal RNA genes, as opposed to everything else. And in the past, it used to be hard to sequence things and expensive, so I did a year as a technician and my undergraduate research sequencing a single ribosomal RNA gene and got a paper out of that. 1,500 base pairs of sequence. And now, of course, we have Illumina machines that can generate 100 quazillion bits of data in half an hour or something to that effect. When this was originally done, you couldn't do, sequencing was expensive and hard. And so increasing the signal from your sample to have lots of ribosomal RNA pieces in your sample instead of sequencing everything from the sample was really important. And you could go use this PCR reaction to make lots of copies of the ribosomal RNA genes, clone those out, and read the sequences of those ribosomal RNA genes from your environmental sample, and then compare them to other ribosomal RNAs, either from cultured organisms or from other organisms in environmental samples. And you can build evolutionary trees of those sequences or do a variety of methods of classifying those ribosomal RNA sequences in order to learn indirectly who was in the environmental sample by classifying their ribosomal RNAs. Just like you might do with leaves or feathers for plants and animals, you can do with fragments of DNA sequence from microorganisms. And there are many subtle steps involved in doing this classification that involve sequence alignments and phylogenetic analysis. That's not probably of anybody's interest right here for these talks, but I'm happy to talk to people about this at some point. This is my year and a half of work to sequence this one ribosomal RNA gene from this chemosynthetic symbiont in 1992. And what this sort of method of going to the environment and sequencing ribosome RNA genes revealed was astonishing. So for example, if you build an evolutionary tree of all the ribosome RNAs that you get from environmental samples, this is a tree made, 2 trees made by Phil Hugenholz. On the left are bacteria, on the right are archaea, and if you classify them into major lineages, what Phil showed here, this is about a 10-year-old paper now, but the point is still the same. There were about 40 major lineages of bacteria at the time. Many of those phyla had no cultured representative. That is, an entire phylum on the tree of life no one had ever studied in the laboratory. They were only known through ribosome RNA sequences from the environment. So we basically, what PCR of ribosome RNA first told us was, we know nothing. That's important to know. I don't want to quote Donald Rumsfeld or anything, but it is useful to know that you don't know anything about environmental samples. But more importantly, what the ribosome RNA sequencing allowed people to then do was ecological studies of environmental samples using ribosome RNA sequences as your marker, just like you would use a list of bird species as your marker, or a list of tree species. And you can do things like estimate the total richness in a sample, the total number of species in the sample, by analyzing ribosome RNA data. You can do biogeography by comparing ribosome RNA sequences' presence and absence across space or time. So ribosome RNA sequencing allowed a revolution in microbial ecology. And it was imperfect. There were many sort of bugs and challenges with doing this, but it opened up a new window into studying microbial diversity by allowing people to compare the taxa across space and time and across different, sort of within a sample over time, and to compare them to everything else on the planet and build these trees. I'm sorry, I don't know if David knows the history of this figure or not. I think it was used in a Les Deflison's paper. And for some reason, they made it anatomically correct in the Nature Reviews paper. I've censored it. I don't think the anatomically correct thing is necessary. What you can then do is do things like classify the organisms present in a sample and compare and contrast them. So for example, in the human ecosystem, as you will hear about probably a lot today, different locations within the human ecosystem have different relative abundances of organisms. If you classify them into major phylogenetic lineages, this is classifying them by the phylum, basically. And the fraction of organisms in those phyla in the different samples varies. And that allows you to ask questions about why you see that variation and do experimental studies. So ribosome RNA in the environment, ribosome RNA from cultured organisms, those continue to march along as studies even while these new eras develop. It's not like people have ignored ribosome RNA sequencing in cultured organisms. The third era in sequencing to study microbial diversity I would consider as genome sequencing. So in 1995, the Institute for Genomic Research, Craig Venter and Ham Smith, sequenced the first complete genome of any free-living organism, Haemophilus influenza. And the method they used was this random shotgun sequencing approach where you blast apart the genome of the organism into random little bits, read those small fragments, and then use a computer to compare the sequence of the fragments to each other and build back together the complete genome of the organism. It doesn't work that simply, but let's pretend like that's all you need to do to read the sequence of the genome of an organism. But that's basically, in essence, with a little 2 or 3 years of follow-up work, in essence, what you do. And this, sequencing the genomes of microbes, led to another revolution in microbiology and understanding of microbial diversity. There were many, many things that were enhanced or even made possible for the first time by having genome sequence data. And it led to the ability to more accurately predict metabolic processes in organisms, lots of use in vaccine and drug design. I've been very interested for years. So I worked at Tiger for 8 years before moving here. And I've been using genomes to study evolution of organisms. The genome sequence, of course, serves as a template for functional studies. Now, this is true for any organism. Genome sequencing revolutionized microbiology in part because the genomes of microbes are small. And you can sequence more of them for the same amount of money than you can for larger organisms. And so now we have, I think, something on the order of 5,000 complete genome sequences of bacteria and archaea in GenBank. And tens of thousands of nearly complete genome sequences. Whereas for plants and animals, there are probably only about 100 sort of well-finished complete genomes of plants and animals. This is just an example. This is a Vibrio cholera. It allowed us to predict sort of the metabolic functions of the organism. The predictions aren't perfect, but they are a template for experimental studies. From an evolutionary point of view, we had hints of what I'm about to tell you before genome sequencing, but genome sequencing really kicked us in the pants about this. So one thing that genome sequencing showed was that if you sequence the genomes of multiple organisms that are considered to be representatives of the same species, so multiple strains of the same species, and compare them to each other, this is comparing 3 strains of E. coli, you find that they share a lot in common. That's probably why we consider them the same species. They share a core of genes that are frequently very, very similar in sequence to each other. But what's weird and troublesome and difficult to deal with is that they have thousands of genes sometimes not present in the other strain of the same species. So E. coli O157H7, the jack-in-the-box, or now the spinach, depends on your generation here, which outbreak you refer to for E. coli O157H7, that has about 800 or 900 genes not present in E. coli K12, the lab rat strain. And that's of 5,000 genes total. Massive genetic differences among very closely related strains of organisms. And although there are many explanations for this, one of the major explanations is that some of these strains are taking up DNA from foreign sources to give them unique islands of DNA, in essence, compared to other things. This lateral gene transfer wreaks havoc on our ability to sort of think about evolution in many ways. Many people have gone so far as to call this a network of life as opposed to a tree of life. However, if you take that part of the genome that is shared among strains, sort of the core of genomes, shared among strains or shared among species, this core is pretty small if you compare lots of species to each other. So if you compare 25 or so species of bacteria, they only share about 80 genes in common. There's not a big core that is shared across all bacterial diversity. But if you build evolutionary trees of that core, you can come up with a reasonable representation of the average evolution of those taxa, sort of the vertical evolution backbone of those species. You might not want to call it a phylogenetic tree. I don't know exactly what you want to call it, but these sort of average genome trees are very useful in the study of bacteria and archaea diversity. And function of organisms does, in part, map onto this tree. So cyanobacteria all group together onto this tree, and virtually all cyanobacteria are photosynthetic, for example. So function doesn't perfectly map onto the tree, but it maps okay in some cases. Gram-positive cell wall maps pretty well onto the phylogenetic tree. Virulence and pathogenicity don't. So there are some things that map well onto the tree and some that don't. So the tree has some useful purposes and some not so useful purposes. As I was mentioning before, the rate of progress of genome sequencing has accelerated incredibly rapidly, in particular for microbes because their genomes are small. So the E. coli genome is about 5 million base pairs compared to 3 billion for humans, and even larger genomes for some other plants and animals. The phylogenetic representation of genomes, even with all these genomes, is not particularly good. So one thing my lab works on is filling in the gaps in the tree of life where there are lineages but no genomes. And I'm happy to talk to people about that afterwards. So the last, do I have until about 9:40 or something? Yeah. So the last era in using sequencing to study microbes is to now sort of do in parallel what happened with ribosomal RNA. So genome sequencing started with cultured organisms in the laboratory where you could get a lot of DNA that was relatively clonal, especially for bacteria and archaea. You can grow up a culture and the different cells in your culture have mostly the same DNA as each other. And so then when you extract DNA and you read random little fragments of the DNA that is actually coming from different cells there, the random little fragments of the DNA overlap with each other because all the different cells are basically identical to each other. However, as I told you with the ribosomal RNA studies, we can't grow a lot of things in the laboratory in pure culture. So we've missed out on using genomics and all the benefits that come with genomics on studying most of the microbes on the planet. This changed sort of slowly over time, but in particular with one set of studies done by a guy named Ed DeLong, who was at the Monterey Bay Aquarium Research Institute. And Ed was really interested in these lineages of bacteria that were found in the surface ocean waters that had been discovered by ribosomal RNA PCR. And by various PCR and in situ hybridization studies and other studies, they had found that some of these lineages represented about 20% of the ribosomal RNAs from almost every surface ocean water sample on the planet. And yet no one had ever grown in the laboratory any close relative of any of these organisms. So here were these organisms that dominated the surface ocean waters, and we knew nothing about them. And so Ed was trying to do experiments in the ocean water to try and figure out, are they photosynthetic? What type of metabolism do they have? And then he started to switch over to using genome sequencing. And pretty much on his first try, he found an amazing thing. So he didn't use random shotgun sequencing. He actually pulled out large DNA fragments that contained ribosomal RNAs of interest. So he knew that these large fragments came from very close relatives to these organisms that had only been seen by PCR, by the ribosomal RNA as a sort of tag. And they sequenced this ribosomal RNA from this bacteria from the surface ocean waters. And they discovered a homologue of a gene called bacteria rhodopsin, which, as the name suggests, it's probably not homologous to the rhodopsin in our eyes, but it works in a very similar way. It's a light-mediated proton pump. And these organisms in the surface ocean waters turn out to have these light-mediated proton pumps. In fact, it's one of the most abundant proteins in these surface ocean water samples is now found. And what this does for the organisms is still not entirely clear. It probably supplements their energy metabolism. They're not fixing carbon, so it's not being used for photosynthesis, but it's probably being used to generate ATP and other cellular energy to do other functions. The proteorhodopsin is sort of pink-purple in color. Many people had been modeling global ocean patterns by looking for chlorophyll, which was the gene, the protein involved in photosynthesis. This one discovery changes the wavelengths of light you need to use for satellite imagery and modeling the energy budgets in the ocean. So when this paper came out, pretty much everybody and their mother who worked on environmental microbiology systems switched to trying to do metagenomics. That's what it's become called, the field of doing genome sequencing from environmental samples. Now today we use random approaches rather than this painstaking mapping individual fragments that Ed DeLong did, mostly because they're cheaper. But the principle is the same. You go to an environmental sample, you extract the DNA, just like with the ribosomal RNA studies, and instead of sequencing just the ribosomal RNA genes, you sequence everything. Now, analyzing this data is a mess. It is very, very challenging. Many of the things coming out of it still right now are dubious in quality because the analysis is so hard. One example of what's hard is you have this mixed community of organisms from complicated ecosystems like soil. Imagine tens of thousands of species in one sample. You randomly shred up the DNA and read little DNA fragments from that environmental sample, and you want to predict what's going on in the ecosystem from this? It's not easy. You want to probably sort the fragments to what organisms they came from. People are still trying to figure out how to do that binning, that sorting. You want to predict metabolic properties, but from a complex community, we don't really know how to do that as opposed to individual organisms. So there's still a lot of mess here, but it is unquestionably revolutionizing environmental microbiology and being done left and right in every ecosystem you could imagine. My lab works on taking those sort of same approaches that were used with ribosomal RNA and applying this to environmental samples, so phylogenetic classification of environmental samples. But there's enormous, because sequencing has gotten so cheap, you can now do ecology with metagenomics by comparing lots of different samples and comparing the metagenomes as opposed to just the ribosomal RNAs for environmental samples. And there are these massive scale projects going on, such as for the human microbiome. There's even something called the Earth Microbiome Project that I am a consultant for, although I'm still not quite sure how they're going to sequence everything on Earth, but they think they are. And they're actually generating a massive amount of data through that project. So I probably don't have time, but I'm just going to quickly point out, I was going to go through the different sequencing technologies. What has happened in sequencing has been unbelievable in terms of the pace at which we can generate data and the cheapness at which we can generate data. The data is not completely easy to use because we get tiny little fragments of data from environmental samples, and the sheer volume of it hurts our computer systems. People now talk about, the file transfer is done by foot as opposed to across the network, because it's sometimes cheaper to copy things to a drive and walk to another site with your 2 terabyte drive and plug it in and release the data than it is to transfer it across some internet systems. But sequencing is getting so cheap that you can imagine using it for all sorts of environmental microbiology purposes. It doesn't mean sequencing is the only thing, but again, it's a nice tool to add in your toolkit to help you do environmental studies of microbes. So I think I'll just end there.
I should probably move on.
Okay. Thank you. And so our next speaker is Dr. Mark Underwood, who is...
We go along, a little bit of an overview. I thought we'd start with some definitions.